Hello, good day everyone. This is your teacher, Mr. Marvin O. Rabino, and today we are going to talk about the journey of the cells as they undergo the process of multiplying a million times, which we call as mitosis. So, what are you waiting for? Let's begin. Well, there are several reasons why mitosis happens in our body. It's not just simply that cells divide nothing. Experience tells us that there must be something that causes something. So there must be a definite reason why mitosis happens. And these are the following reasons. Number one, and that is for growth. Number two, that is repair or healing of damaged tissues. A good example for that one is when we have a cut in our blood vessels, or shall we simply say wound in our body. It's actually mitosis that is working there. That's why your wounds heals. It's not the medicine that actually heals your wound, but the medicine just helps the healing process by killing the bacteria that surrounds the wound. But it's actually mitosis that acts as a mechanism in order to heal your wound and to connect once again the damaged tissues. Next, we have a sexual reproduction. Well, you might think that a sexual reproduction only happens in plants, but basically you are correct because sexual reproduction is very common in plants, but sexual reproduction means that one cell can actually produce multiple number of cells from just a single cell. So that is what a sexual reproduction means here. Now, you have to remember also that some cells in our body divide constantly. A good example for this one are of course the cell embryo, the skin cells, and as well as the epithelial cells in your body. But there are cells that rarely divide or they never divide. Good example for this one is your brain cells in your nerve cells, spinal cord cell, and of course you have your cardiac cell. That's why, as you notice, as time passes by, when you grow old, some of these cells can never be replaced again because simply they stop dividing or they rarely divide at all. So no wonder that our life will always perish as time goes by because our cells, or most of our cells in our body, rarely divide or they never divide at all. Now, in cell division, you have to remember, we have to go back to the basics that there are two kinds of cell division. Namely, we have the mitosis, which we'll be talking about in this chapter, which is the division of the somatic cells. And we have the meiosis. This is another type of cell division. But in contrast with mitosis, this does not involve somatic cells, but instead, meiosis involves your sex cells. Specifically the creation of new sex cells. Of course, you know the sex cells in our body. So we have an example here. In male reproductive organ, we have the sperm cell. And in the female reproductive system, we have the egg cell. And of course, we know the story that when Romeo meets Juliet, they form their destiny. And that is the embryo. And before we are going to deal with the basics of cell mitosis, there are just some key terms that you need to understand so that by the process of the discussion that will happen in the next slides, you will not wonder anymore what do these words mean or how does these words are used in the study. So let's begin. First, I'm going to introduce to you what are somatic cells. Simply, they are just body cells or cell whose genes will not be passed on to future generations. A good example for this one is your muscle cells. Of course, muscle cells are called meiotic cells. You have also your cardiac cells. It's basically the composition of the cells that are found in your heart. You have also our epithelial cells in our body. Basically, these cells will not be passed on to the future generation, because for each generation these cells are actually different from one offspring to another offspring. Next, we have the germ cell. Basically, they are not germs, that's what you think, but just called germ cells because this is a cell that is destined to become a gamete. When you say gamete, it could be an egg cell for female or sperm cell for male. Or these are cells whose genes can be passed on to future generations. Well, a good example for germ cells are, of course, your sperm cell and your egg cell in your body. So it's basically very understandable. Next, we have the diploid. I will denote diploid as 2N. Now, 2N here means that a cell with 2 chromosome sets in each of its cells. A good example for this one are all the body or somatic cells. Now, basically, class, this means that when you say that it is a diploid, that means it contains the complete number of chromosomes that a certain living thing possesses. Let's say, for example, a human body cell has 46 chromosomes. That is our diploid number of cells. That means it contains 23 pairs of chromosomes. Now, since they are pairs, just multiply it by 2, 23 times 2, then our total chromosomes in our body is 46. Remember that. That is diploid. Next, we have the haploid, or I will denote that one as N. Or this means that a cell with 1 chromosome set in each of its cells. Example for this one are all the gametes that are found in the male or female. Let's say for males, of course, they are called the sperm. And for females, they are called the egg cells. So basically, it's like this. The haploid contains half the number of chromosomes that an organism possesses. Let's say for human beings, our complete number of chromosomes is 46. So if we are going to divide it by 2 because it's a haploid, that means all the gametes of human cells contains 23 chromosomes. Of course, that corresponds to our sperm cell and to our egg cell. Next, you have to also to understand some of the characteristics that are only unique to mitosis. Say, for example, in mitosis, a diploid cell will always give rise to a diploid cell. That means one cell, let's say, for example, in human beings, one cell with 46 chromosomes. Remember that 46 is our diploid number of cells in human. When they undergo mitosis, when they divide, they will still produce a diploid cell which contains exactly the same number of chromosomes, which is basically 46. Next, the chromosome number remains the same as what I told you a while ago. So basically, it's very understandable. Next, the DNA remains identically the same. That's what is unique with mitosis. Even though they undergo division for several times, the DNA will always remain identically the same. And one cell with a diploid number of chromosomes will always gives rise to 2 cells. That means one cell, when they undergo cell division, will always produce 2 cells with diploid number of chromosomes. Now, you also need to understand that there are actually processes that are involved in the preparation for your cells before they undergo cell division. So, A typical cell goes through a process of growth, development, and reproduction, which we basically call as the cell cycle. And most of the cycle is called the interphase. As you can see in the diagram or in the picture, interphase is the longest phase in the cell cycle, which comprises of 3 stages, namely the gap1 or the G1 phase, the S phase or the DNA synthesis phase. We also have the final stage of interphase we call as G2 or gap2 phase. Now, there is a special case or there is a special phase in the cell cycle wherein some cells cease to divide. That means instead of going through the whole cycle again, they stop shortly after the cell division or the M phase, mitosis. That means after mitosis, they stop dividing. And that is indicated by an arrow pointed outside of the circle in our diagram. We call that special case as the gap 0 phase, or we refer that one as the G0 phase. Now, as what I've said a while ago, the longest phase in the cell cycle is the interphase. This is the stage where most of the cell's life are spent. 3 stages of the interphase are the G1 phase or the growth 1 phase or the gap 1 phase, as what the other books call it. The next is the S phase or the DNA synthesis phase, and last is the G2, or we call that one as gap 2 phase. Now, as you can see in the diagram, G1 phase is responsible for the continuous growth of your cell. This is the time where or when your cells grow, develops its organelles, and develops all its parts. So basically, it's just like when we are still a baby that we develop our muscles, we develop our organs inside our mother's womb, and we also develop our sense organs, for example, our eyes, our ears, our noses, etc. So it's just basically like growing up. So basically your cells also have to undergo growing process, and that is in G1 phase. Next, after growth 1 phase, soon as your cells are ready to live or mature enough to enter the S phase then follows, wherein S stands for synthesis. That means in this phase, it's time for your cells to undergo DNA replication, wherein the DNA is actually duplicated, which will be utilized later on in the M phase or the mitotic phase. Next, before leaving the S phase, there is actually a checkpoint located just before G2, and we call that one as the S phase checkpoint. Now, that checkpoint will actually check whether all the DNA of the cells are duplicated and that it is properly duplicated. By the time that they meet all the requirements before entering the G2 phase, then they can now enter the last stage of the interphase, which we call as the G2 phase. Now, in G2 phase, it's actually the final preparation for cell division. It is when your cells grow continuously and prepares for cell division. And of course, after they meet all the requirements of the G2 phase, it is now time for your cells to undergo mitosis. We basically call mitosis as the mitotic phase of the cell cycle. Now, as you notice that there is an arrow that is actually pointed outside of the circle, that is a special case because shortly after the M phase or the mitotic phase, some of your cells will actually cease to divide and rarely divides at all. A good example for this one is your spinal cord cells. Of course, they are located in your spinal cord. You have your brain cells. You also have your cardiac cells. So basically, these cells will actually cease to divide after entering the M phase. We call this phase as the G0 phase or gap 0 phase. That comes the whole process of the cell cycle. Now that we know the whole process of cell cycle, let us now talk about the topic that is focused today, which is all about mitosis. Now, you have to remember that all living cells come from other living cells. That means to say that a cell actually comes from an ancestor cells. That means every cell in our body actually came from the previous cells that actually died inside our body. And that during mitosis, the nucleus of the cell divides, forming two nuclei with identical genetic information, or shall we say, identical DNA. That means one cell in the diagram undergoing DNA replication will actually undergo mitosis phase, wherein its nucleus divides, and then finally, after the nucleus divides, one cell will now give rise to two daughter cells with exactly the same number of DNA and identical genetic information. Now, in mitosis, it produces two genetically identical cells, and that it follows step-by-step process, which we call as the prophase, metaphase, anaphase, and the telophase. So as you can see in the diagram here, that is just the schematic diagram of all the processes that happens during the M phase or the mitosis phase. Now, I'm going to discuss this one by one, so you have to prepare your notes, take down the important information that are stated in this slide, and that you also have to take note all the significant events that happens in each of these phases. So, let's begin. Let's have the prophase stage, which is basically the first stage of the mitotic phase. Now, you have to remember that in this stage, cell begins the process of cell division, and that in this phase, you have to take note properly that the chromosomes condenses. So, let me give you a clear picture for this one. In the diagram or in the picture shown, in letter A, this is what your DNA looks like before it enters the prophase stage. It is the uncondensed form of your DNA. So, it somewhat looks very messy. But in the prophase stage, your DNA starts to condense into duplicated chromosomes. Now, how are these chromosomes are condensed? It is actually because of the action of the protein that is present in your cell, which we call as the histones protein. It is indicated by the green molecule in the picture. Now, these histone proteins will help the DNA coil your DNA into a packaged DNA, which we call as the chromatin. Now, as the chromatin folds up, it becomes the chromosomes. So, this is now your duplicated chromosome, which will be shown up in your prophase stage. It's basically a double-stranded chromosome. Now, in the prophase stage also, you have to remember that the nuclear envelope, which actually surrounds the nucleus of the cell, will start to disappear. And this is how it looks like in the microscope. Now, another important event happens in the prophase stage, wherein what we see as something insignificant organelle in the cell actually plays a vital role in cell division, and we call these organelles as the centrioles. Now, in the prophase stage, centrioles migrate to the opposite poles of the cell. Now, centrioles will actually be responsible for the division or the movement of the chromosomes to the opposite poles of the cells in the later stage of the mitotic phase. But for the meantime, we have to emphasize that asters and spindle fibers are also formed in this phase, as indicated in this diagram, where the asters are the green colored particle in the picture, and the spindle fibers are the red lines connecting the chromosomes in the diagram. If you're going to illustrate the prophase stage, basically looks like this. Well, this is just a drawn diagram, but simply you just can understand this type of illustration, then we can upgrade it into a more detailed picture. It basically looks like this. So as you can see, you have the centrioles, you have the spindle fibers forming, and as well as the asters that extends outward of the centrioles, and we have the sister chromatids, or basically the duplicated chromosomes. Now, after the cells enter the prophase stage, it is now ready to enter to the next stage. The next stage as the metaphase stage. Now, in the metaphase stage, it is just actually a very short event in the mitotic phase. So, basically what happens here is the chromosomes, which is basically present in the prophase stage, your duplicated chromosomes will actually line up at the equator of the cell, and we call this equator as the metaphase plate, with the centrioles, which are formed during the prophase stage, at the opposite ends, and the spindle fibers will now begin to attach themselves to the centromeres, as indicated by the illustration here. As you can see, the spindle fibers begin to connect to the centromeres of your chromosomes. By the way, the centromeres are the center part of your chromosomes that connects the duplicated chromosomes together. Now, this is actually the metaphase stage as viewed from the microscope. Now, as you can see on the left portion, the two left pictures, they are actually the metaphase stage of the Allium cepa plant, or basically the onion plant. While here, on the right picture, is basically the animal cell metaphase stage. So, wonderful, isn't it? Now, if you're basically not too observant in identifying what metaphase stage looks like, then we can just simply illustrate it this way. Basically very understandable. You're just going to line up the cells at the equator of the cell, or the metaphase plate. Now, after the cells line up at the equator of the cell, or the metaphase stage, or the metaphase plate, that is metaphase plate, it's now ready to enter the anaphase stage. Now, in the anaphase stage, it is actually where your centromeres divide, or basically it's the start of the division of the duplicated chromosomes, or the sister chromatids, wherein, as you can see on the diagram, each chromosome goes from having two sister chromatids to being two separate chromosomes, and that these two sister chromatids, which are basically connected before the anaphase stage, will now separate and move to the opposite poles of your cell. Now, what makes this possible is the action of the spindle fibers, because spindle fibers here contract and the chromosomes are pulled to the opposite poles of your cell. So as you can see here in the picture, this is actually a very, very vivid picture of the anaphase stage of the Allium cepa plant, or basically the onion plant. Nice, isn't it? Well, if you plan to further have some experiments regarding Allium cepa plant, well, you can actually do it yourself. The mitotic division in Allium cepa plant is actually very visible under a compound microscope. Now, simply you're just too not too eager to look at the microscope to identify what stage of mitosis is referred to by the scenario, then we can just actually simplify anaphase stage by this illustration. Basically, you're just going to draw chromosomes separating and moving towards the opposite poles of the cells, and then we have pictured here that the spindle fibers, which are actually indicated by black lines, pulling the chromatids towards the opposite poles of the cell. Now, by the time that the sister chromatids separate at the opposite poles of the cell, telophase begins shortly. Now, in the telophase stage, the nucleus actually divides, and that the chromosomes are now at the poles of the cell. Before I begin with telophase, or before I discuss further with telophase, by the way, telophase is the division of the nucleus. We call also that process as the karyokinesis. Now, the nuclear envelope, which was absent during the prophase stage, will now begin to reform around the two sets of chromosomes. So this is actually the picture of how the telophase stage looks like in the plant cell. So this plant cell is just basically the same plant cell I've been talking a while ago. This is the onion plant. Now, just simply not, again, you're not too very observant with the microscope and you just want to simplify everything, then you can just simply illustrate it this way. Just cut the cell into two parts, but basically not divide it, not separating the poles of the cell, just simply going to have a boundary line between the two poles. Then that's basically how telophase looks like. Now, if you think that this story is over, my friends, well, basically it's not yet the end. It's just the beginning of another story, because in the telophase stage, which is the last phase of mitosis, your cell does not actually divide separately. It's just their nucleus that are actually separated during that stage. Now, there is a special process that is needed in order to completely separate the cells differently from each other, and that is what you call as the cytokinesis. Basically, class, cytokinesis is not included in the M phase, but it's another process in your cell cycle. But there is a need for us to include cytokinesis in the picture, because in the cytokinesis, this is actually where your cells will divide separately into two identical daughter cells. Basically, cytokinesis is just the division of the cytoplasm. Let's go back to our previous knowledge about the parts of the cell. We know that the cytoplasm is the jelly-like structure in your cell, which actually most of your organelles are suspended. Now, cytokinesis is very important because it actually divides your cells separately into two identical daughter cells. Now, in plant and animal cell, cytokinesis actually happens differently and uniquely. For instance, in the animal cell, a cleavage furrow forms and separates the daughter cells. That means to say, in animal cells, there is what we call as the cleavage furrow formation. As you can see in the diagram here, this is actually how a cleavage furrow is formed in a dividing frog cell. So basically, the center part there, where there is somewhat a Pac-Man formation, is actually the cleavage furrow of the dividing frog cell. Now, in our cells, or rather in plant cells, a cell plate formation indicates that cytokinesis happens. And the cell plate formation is actually responsible for the separation of the plant daughter cells. Basically, it's like this. So, as you can see in the plant cell, of course, it's basically composed of cell wall and plasma membrane, and it's actually the vesicles that are found in your plant that actually helps form the cell plate of the plant. And the formation of this cell plate will actually help the cells in your plant separate into two identical daughter cells. So, in summary, this is actually how cell plate formation happens in a plant cell. Basically, this is just the allium cepa cell plate formation, which is basically the onion plant. Remember, allium cepa is just the onion. Don't forget that. Now, in summary, in the plant and animal mitosis, you can summarize them as follows. Now, in animal cell, the centrioles and asters are present. The daughter cells are separated by the cleavage furrow. And, contrast to the plant cell, in the plant cell, take note that there is no visible centriole or aster. But the daughter cells in the plant cell are separated by cell plate, or the cell plate formation. Now, let's try to test your understanding of the different processes of mitosis. So, to begin with, I have sets of pictures here wherein you are going to identify the stages of mitosis referred by each picture. Are you ready? So, let's start with number 1. What stage of mitosis is referred to by the picture 1? You can pause the video and think for a while. You have the answer already? Well, you got it. It's actually the prophase stage. How about the second picture? Pause the video and think. So, let's see if you got the answer. Well, you're basically correct. That is the metaphase stage. Now, let's go to the third picture. Pause the video and think. Well, let's see if you can still recall the different processes of mitosis. Got it correctly. That is the anaphase stage, because basically it involves the separation of the duplicated chromosomes or the sister chromatids. Next, we have the fourth stage. What is referred by the picture number 4? Well, you got it correctly. That is the telophase stage. And last but not the least, how about picture number 5? Can you guess what process is referred to by picture 5? Well, you are correct. That is actually cytokinesis. Give yourselves a big round of applause. Now, to sum it up, these are the basic stages of the cell cycle. Just remember the acronym IPMAT-C, which basically stands for the interphase, prophase, metaphase, anaphase, and the telophase. And let's not forget that C stands for cytokinesis. And if you simply found it boring to have this acronym, you may adapt these other mnemonics to easily memorize the stages of the cell cycle. So, say, important people must analyze tasks correctly. Basically, I stands for interphase, P for prophase, M for metaphase, A for anaphase, T for telophase, and C for cytokinesis. But if you simply don't like this acronym, you may adopt this alternative mnemonics which I developed personally. Ready? Impatient people may attack teachers constantly. So I hope that you enjoy this discussion of today. Thank you so much for listening, and may God bless you all. For the next meeting, we are going to have a short quiz regarding our discussion for this afternoon's topic. Please prepare for a quiz for our next meeting. Thank you and goodbye.